A visual isotope effect, the chromium-6 oxidation of 2-propanol. This demonstration uses time-lapse photography to compare the chromium-6 oxidation rates of 2-propanol and 2-propanol D7. Experimental design. The alcohols are added in rapid succession to each of the 2 vials containing the light orange chromium-6 reagent. A third vial, at left, contains no alcohol and serves as a control. A color change is used to monitor the reaction rate. The light orange chromium-6 is reduced to chromium-3, which is blue. On the next slide, you will be able to see that chromium-6 has begun to react with the alcohols. This first shot was taken approximately 30 seconds after the reactions began. Therefore, 0 seconds is when the photo started, but not the reaction. Here is the first shot of the experiment at approximately 30 seconds after the reactions began. We ran the experiment for 90 minutes and took a photo of the vials every 10 seconds to document the reactions. We have stopped the time-lapse at 1000 seconds to show you that the middle vial with 2-propanol has completed reacting, while the vial to the right with 2-propanol D7 continues to react with chromium-6 for the next 4400 seconds of the experiment. For your convenience, we have sped up the remainder of the time-lapse, and at its conclusion there will be a brief explanation of what is happening in the vials. The sample containing 2-propanol D7 reacts more slowly than the vial containing 2-propanol. Now you might be asking yourself why this is the case. Well, the difference between the 2 alcohols is the presence of deuterium, one of the 3 hydrogen isotopes. With 1 proton and 1 neutron, deuterium is twice as heavy as protium, the most abundant hydrogen isotope. The larger mass of deuterium causes C to D bonds to be shorter and stronger than C to H bonds. This difference in bond strength is due to a mass-dependent quantity known as zero-point vibrational energy. If a reaction rate is thought to depend upon the rupture of a C to H bond, then this hypothesis can be tested by replacing H with D and seeing whether or not the reaction rate slows. The oxidation mechanism consists of 2 processes. The first is formation of a chromate ester. This process consists of several discrete reaction steps typical of acid-catalyzed esterifications. The second is the actual oxidation-reduction reaction. In this step, a C to H bond is broken and a pair of electrons is transferred from 2-propanol to chromium. 2-propanol is oxidized to acetone and chromium-6 is reduced to chromium-4. Chromium-4 is a reactive species, which undergoes further reduction to chromium-3. Which process determines the overall rate of the reaction? Because the deuterium-containing alcohol slows the reaction rate, this suggests that the second step is rate determining, or the slowest step of all the processes. The other steps are rapid by comparison. This is because the second process is the only one in which a C to H bond is broken. The rate slows because the stronger C to D bond is more difficult to break. This is called a kinetic isotope effect. Chemists can use information like this to understand how chemical reactions work. Researchers in the late 1940s, in fact, used the approach demonstrated here to confirm the slow step of the chromium-6 oxidation process. They determined that 2-propanol-D reacted about 6 times more slowly than unlabeled 2-propanol. We use 2-propanol-D7 in our demonstration because it is less expensive than 2-propanol-D. The two CD3 groups in 2-propanol-D7 do not contribute much to the isotope effect, so the two labeled compounds behave nearly the same. In conclusion, time-lapse photography can be used to demonstrate the different chromium-6 oxidation rates of 2-propanol and 2-propanol-D7. Because the isotope effect in this process is large, the effect of deuterium incorporation in this system is readily visualized. This slide shows the sources that were used in this presentation. A visual isotope effect was prepared by Chloe Soltis and Professor Dan O'Leary of Pomona College. We thank the Pomona College Chemistry Department for funding this project.